Hello and welcome to our last lecture of the course Introduction to Mechanobiology. So, in last lecture over the last few lectures we are discussing about various tools in mechanobiology. Okay. Among the tools I introduced hydrogels, okay, AFM, okay, assays like traction force microscopy or laser ablation or trypsin deaddition, okay, for probing the contractility of cells, okay, AFM for measuring stiffness of samples, for imaging topography, okay, for also doing adhesion measurements, etc. Okay. And the last class spoke about soft lithography, okay. And using this you can regulate cell shape, or you can use micropillars, or you can fabricate micropillars for traction as an alternative to TFM. Okay. So, I had briefly talked about one study which linked cell shape or cell spreading area with apoptosis, okay, dictated by the extent of cell spreading. Okay. So, similarly there are multiple other studies which have focused on using microfabrication to glean insight into molecular processes. So, one other study by Doyle et al., okay, published in J Cell Biol, okay, demonstrated that 1D migration is similar to that in 3D on fibrillar surfaces. Okay. So, by fibrillar matrices I mean aligned collagen matrices or similarly. So, how did they go about doing that? So, what they did was they patterned these lines which varied in their width W, okay, and they tracked. So, you have these lines, you plate cells on top of these lines, okay, and you track their migration, and what they observed, okay, what they observed as a function of width. Okay. So, what they observed as a function of width was as the width increased, okay, as the width increased there was a drop in the extent of migration. Okay. In 1D lines, okay, as your increase in width your speed dropped. Okay. I do not want to discuss this study anymore, but I refer you to this paper. It would give you an insight as to how cells migrate on fibrillar matrices. So, today I would introduce another tool in mechanobiology. Okay. So, this is called fret. So, what is fret used for? So, fret is used for probing protein-protein interactions. Okay. So, essentially the question is, how do you show that a protein A binds to protein B, or like a receptor ligand interaction. Okay. So, if you use imaging, so all you would get information about co-localization, but this is not enough to say A and B interact, because we know, for example, at focal adhesions you have greater than 100 proteins which localize. Okay. It is not that each of these proteins is interacting with each other. Okay. So, one way to go about it to find out the interaction of two proteins is using co-immunoprecipitation. Okay. So, what is done in this approach is you collect the cell lysate, let us say, and incubate with beads, okay, which are coated with antibody against A, let us say. Okay. So, we are proving the interaction of A and B. So, these beads will get rid of all those protein aggregates where A and B are attached to each other. Okay. Then you purify this component. So, you purify, you run on a gel, and then you can probe it with the antibody to B to see if you get a band. Okay. So, this is of course a useful technique, but what this technique does not give us information is about dynamic interaction. So, because it depends at what time point you are collecting your lysate. Okay. But if this interaction is dynamic, let us say at different stages of time or spatially regulated, then you will not get this information, because by collecting this you are getting an average response. Okay. This is where the technique of fluorescence resonance energy transfer is useful. Okay. So, FRET is short form for fluorescence resonance energy transfer, okay. And this involves the transfer of energy from an excited donor, okay, donor fluorophore, to an adjacent acceptor fluorophore. Okay. So, generally what is commonly used is CFP and YFP. These are all variants. Okay. So, these are color variants of green fluorescent protein. Okay. So, what you have is for these two proteins, these two fluorescent follicles, CFP and YFP, your excitation, okay, is at 440 nanometers, emission is at 480 nanometers for CFP. Okay. But the excitation peak for YFP is 510 nanometers, okay, and the emission is at 545 nanometers. Okay. So, now what you can do, so what has been showed, okay. So, so if you have during the lifetime of excited state of CFP, okay, an energy field is gets created, okay, by oscillation of the excited electron. Okay. Now, YFP, so this energy can then be used to excite YFP, okay, provided, so there is one clause, the distance is very low. Okay. Now, how low is low? So, what has been shown that the efficiency of this transfer of energy okay, you can have this okay, decreases as r to the power 6th power, okay, which means if r is the separation between these two fluorophores, your energy will decrease or reduce as r to the power 6. So, you have an efficiency, FRET efficiency okay, which falls off really fast, and this fall off. So, let us say beyond 60 nanometers, 60 angstrom, your freight efficiency is close to 0, okay, 60 or 70 angstroms. So, essentially your floor to floor distance has to be less than 70 angstroms to be detected by this method. Okay. So, now what do you do in this freight tech? Okay. So, if you have two tagged proteins, let us say, okay, protein A which is tagged to CFP, protein B which is tagged to YFP. Okay. So, in freight what you do? You excite at 440 nanometers and you collect the emission spectra of 545 nanometers. Okay. So, this is emission of YFP and this is excitation of GFP. Okay. So, this freight efficiency will work. Okay. This freight efficiency is good when these two molecules are very close enough of the order of 60-70 angstroms or smaller. So, if they are so close, then you can safely make an assumption that these two proteins are really interacting. Okay. So, having said that, I would next come to how can we make use of freight, okay, for mechanobiology studies. In that regard, I would like to discuss this particular paper, Grashoff et al. This is a Nature paper, 2010 paper. Okay. So, by now through this class you have got to know about this protein called vinculin. Okay. So, vinculin is a FA protein, and if you were to look at its structure, okay, you have a head domain and a tail domain which are connected by a flexible linker. Okay. In the head domain you have binding partners like tailin, alpha actinin, okay, and in the tail domain you have actin, okay, as one of the binding partners. Okay. So, these head and tail, okay, let us say VH corresponds to the head domain and VT corresponds to the tail domain. Okay, they are separated by a flexible linker. Okay. Now, binding of VH, okay. So, when VH binds to tailin, okay, this event leads to recruitment of vinculin to focal adhesions, okay, and VT as I said binds to actin and paxillin. Okay. Now, what people have found that force is an important determiner of vinculin localization at focal adhesions. Okay. And vinculin deficient cells, okay, they display faulty spreading and migration. Okay. Also, vinculin deficient cells are softer compared to controls and they also exert lesser forces. Okay. Now, vinculin is also assumed to be an important part of the molecular clutch. Okay. And vinculin is known to colocalize with areas of high forces during leading edge protrusion. Okay. So, based on all of this, people have thought that vinculin participates in strengthening additions. Okay. Such that if there is an increased force, then you have increased recruitment of vinculin. So, focal adhesions will grow in a way that force per area is constant. Okay. This we discussed that paper which showed that you have on an average of 5.5 nanonewton per micron square of focal adhesion. Okay. So, they wanted to understand what happens to the structure of vinculin when you expose it to force. Okay. So, in this regard, okay, so they made use of a construct, okay, which they named as the tension sensor. Okay. So, what is their tension sensor design? So, what they did, so you have the tail domain of vinculin, okay, the head domain of vinculin, okay. Let us say PT, you have, okay, so this is a fret pair, okay, and we say linked using an elastic linker. Okay. So, if so this is the tail domain, so the head and tail domain are connected by this construct or a tension sensor which has an elastic linker in between. Okay. So, if the elastic, the idea being that if the elastic domain is stretched, okay, so when you have stretching of this elastic domain, okay, in this configuration your fret signal is going to go low. Okay. So, this is the idea of their tension sensor design. So, what they first did was they wanted to evaluate the responses of this vinculin to cellular forces. Okay. So, for that they first generated a vinculin minus cell, so which does not have any vinculin of its own, and then they transfected it with this vinculin tension sensor is the construct. Okay. And what they showed was when they plated these cells, and what they were looking at is the fret index or the fret efficiency. Okay. So, when cells were plated on polylysine, whatever be the fret index, okay, when they plated it on fibronectin, the fret index dropped. Okay. So, this was one of the big things. So, on fibronectin the cells were spread. Okay. So, these results suggest, okay, that fluorescence, under force. So, you know that once the cells spread out, they will exert traction forces and so on and so forth, that should lead to a decrease in the fret index. In polylysine surfaces, cells were much more rounded, and consistent with this the fret index was much higher. Okay. You could also track the lifetime. So, they tracked the fluorescence lifetime of this, and they found the lifetime, okay, of the tension sensor. Okay. This is my lifetime, how long they exist at focal adhesions. This was much longer compared to a control construct, okay, which they call as WinTS, okay, sorry, WinTL. So, contracts was WinTL, where it only had the tail domain connected with the fluorescent construct. Okay. So, this was the first demonstration that when you plate on ECM coated substrates, this vinculin is under tension. What they did next, okay, was that to demonstrate whether vinculin recruitment to focal adhesions is force dependent or not. Okay. So, whether recruitment correlates with force transmission, what they did was they took cells, okay, and treated with two contractility perturbing drugs. One is Y27362, which is the ROCK inhibitor and they used myosin 2A knockdown cells. Okay. So, in both these treatments your contractility should go down. So, if the contractility goes down, then the force at adhesions is supposed to go down. Okay. So, consistent with this idea, what they found was when they tracked the fret index. Okay. So, if this was the average fret index when you have the tension sensor construct under control conditions, when they knocked down 2A or Y27632, so in both these cases the fret index went up. Okay. So, the fret index went up means that the extent of stretch of the elastic linker decreases. Okay. So, this shows that there is this association between forces and its localization during addition. Okay. They extended the study to ask that how does vinculin localization or tension in vinculin vary during cell migration. Okay. And for this purpose what there was, they tracked the fret signal from the protruding side of the cell from protruding adhesions and retracting adhesions. Okay. So, as you recall that in the molecular clutch, after the cell exerts the actin dependent protrusion forces, you begin to have the formation of these focal complexes at the leading edge. Okay. Similarly, at the trailing edge you have these adhesions which will break because of contractile forces at the rear. Okay. So, what they found was, compared to the protruding edge and the retracting edge, when they compared the fret signal, they found that at the protruding edge the fret index was lot less compared to the retraction edge. Okay. So, this shows that higher fret index means lower force. Okay. So, to look at this in greater detail, what they did was they correlated fret index with focal adhesion size. Okay. And they found at the protruding edge, so they have two axes, one is your fret efficiency and one is your focal adhesion size. So, they found as a function of time, if the fret index increased, the focal adhesion size also grew. Okay. So, this was at the protruding edge. At the other edge, what they found, so this is positive correlation in this case. In the other case, what they found was the fret index increased, but the focal adhesion size is decreased. Okay. So, these suggest that vinculin recruitment and force bearing are two separate events. Okay. So, this is a major difference in the two separate portions of the cell. Okay. So, this concludes our discussion of fret. Okay. So, in the past few weeks, we have discussed various papers. I would like to list what are some of the reading assignments that you would have to do for the last 1 week. So, first of all, read the Pelham and Wong paper. Okay, which linked stiffness and spreading. Okay. The biophysical paper. Okay. So, it showed the collective influence of ECM stiffness and ligand density on spreading. Okay. Our Low et al. Paper, this was again grouped by Wong. Okay. So, this demonstrated, sorry, it is 2000, demonstrated durotaxis. Okay. Okay. This is a 2000 again Biophys J. Sorry, this is Journal of Biomechanics. So, this is a review paper, okay, which discusses how you can combine optics and mechanobiological tools for probing mechanobiology. Okay. This is a paper introducing trypsin deaddition as an assay for probing cell contractility. Okay. You have this is a 2005 paper which introduced laser ablation for probing contractile mechanics. And the paper I just discussed, okay, on vinculin tension sensor. Okay. So, this concludes our course. I hope you have enjoyed this course, and I welcome you to read many of the papers which are listed in one or more of the papers that I have suggested as reading assignment, where you have references to many other papers which discussed some or other mechanobiological contexts relevant to cell behavior. Thank you for your attention.